Oh yes, thank you so much for having me. Yeah, it's great to be here. So, well, be here, I guess, like as a manner of speaking. Yeah, so I am Sam. Obviously, it is not July 7th, 2020, unlike what it says on the slide. But I am the founder of the Applied Biotechnology Laboratory at the Crick Institute. We started we started in January, this past January, so January 2021. Have just been getting up and running. Basically, I'm a technologist. I did my PhD at MIT and then moved, rather than doing a kind of standard postdoc, I went to be an entrepreneur in residence at Petrie, which is a biotech incubator in Boston, and worked with them on founding two different companies based on technologies that I've invented. So I have a little bit of experience both on like the academic side and also on the entrepreneurship side. And so I've now started this lab at the Crick, and basically what our goal is to do what I call like need-oriented biotechnology, which is to say like developing biotechnologies, but like with a view towards how those technologies are actually going to make it out into the world, either into other academic labs or into a clinical setting, and basically achieve concrete impact by collaborating with investors, pharma partners, people like that. So just putting a very strong emphasis on not just the development, but also the deployment. And we basically have 4 different areas that we're working on right now. So we're working on connectomics. So 2 different research tools, basically. So connectomics, wanting to work towards something that would be like a genome project for the brain. On the one side, on the other side, like contextual transcriptomics, so spatial and temporal transcriptomics, which is what I'll tell you guys about today. But then we're also working on some more like technologies with like clinical applications. So things like we have a liquid biopsy technology that we're developing in the lab for detecting proteins at ultra low concentrations in the blood. We're also developing some new viral vectors for gene therapy. And I would be remiss not to say that we have fully funded postdoc positions open. So if you listen to this talk and you're looking for a postdoc and it sounds exciting to you, and you want to publish high impact papers and build technologies and start companies or whatever you want to do, please, by all means, reach out to me. I would love to hear from you and chat. But so I think I was invited here basically to talk about Slide-seq. I think listening to some of the talks and also listening to, anticipating the talks that are going forward, Slide-seq at this point seems like this story is now 2 years old, which in this climate seems like a long time ago. But Slide-seq, I guess, was a technology that I invented is a technology that I invented along with Evan McCosco and Fei Chen at the Broad Institute, along with my co-first author, Bob Stickles. And I should say just at the beginning that actually technically my PhD was with Ed Boyden at MIT, and Ed is not actually an author on the Slide-seq paper. And so I always just like to acknowledge the fact that Ed was very happy with me going off and working with Fei and Evan, even though he was paying for my PhD and he didn't contribute to the paper, and that was fine with him. So I really appreciate that. I always just like to point that out. But so Slide-seq was basically, we developed it as the first spatial transcriptomic method with a single cell spatial resolution. And the idea, which is, I feel like this is not a story that I really need to tell or like this is not an intro I really need to give. But at that time, right, which was I guess like 3 or 4 years ago when we started working on it, there were kind of 2 paradigms for looking at gene expression with spatial resolution. The first would be in situ hybridization methods, which like MERFISH over here, or StarMap, are methods where you basically have to like choose the transcripts that you want to look at, and you can then look at those transcripts with like very, very high spatial resolution, right? So like single transcript resolution. But then the problem with those methods is that those methods end up they're very hard to implement in practice. They usually require a lot of tissue-specific optimization, and you have to know in advance which transcripts you want to look at. On the other side, are the sequencing-based methods. So, for example, shown here is the original spatial transcriptomics, which at that time got very, very low resolution. And as we're now hearing, those methods now have very high resolution. But just to kind of give the background, at that time there was very low resolution. So our idea was basically, how do we bridge the gap between these 2 paradigms and find a way of doing transcriptome-wide gene expression mapping with a single cell spatial resolution. And the idea was basically just to take inspiration from single cell, droplet-based single cell RNA sequencing, where you encapsulate cells in droplets with microfluidic beads and transfer the RNA from the cells onto the beads, and then barcode them that way. And basically we just said, what if rather than having each bead represent a cell, instead each bead would represent a location? And so what we did was that we took the beads, we deposited them onto the surface into these arrays called pucks. We then, and so this is on the left basically is a cover slip. It's a 30mm glass cover slip where you can zoom in and you can kind of see the beads in each individual array. There will be like a 100,000 beads in each array, and then you can see them kind of just on the surface there on the right. The beads are 10 microns. Right, which in the context of Stereo-seq, for example, sounds really large. That is a totally arbitrary number that we chose because they were the smallest that we could buy commercially. You can actually make smaller beads, no problem, if you want. I think that at this point, the problem of spatial resolution in spatial transcriptomics is probably solved. And so we decided to let other people solve that problem. But so in traditional Slide-seq, the beads are 10 microns. You then go through and you decode. So the beads are deposited randomly. So you can then go through and basically we built a custom sequencer to go through and decode the barcodes. So for example, this is an image of some Slide-seq beads on a surface, and you go through and just round after round, you can see different, the base calls associated with each of those beads. And so then you build up at each location, you build up a barcode, right, associated with the bead that's at that location. You then take your tissue, you slice it onto your array, and you do some standard wet lab steps, which are very similar to other methods that are now being published. You can then go through and match the barcodes that you get a library, which is essentially equivalent to any library that you get out of single cell RNA sequencing. But then you can go through and you can match the barcodes in that library with the barcodes that you get out of the barcodes that you get out of the spatial sequencing. And so, because the data is equivalent to single cell RNA sequencing data, you can run all the kind of clustering on it that you would normally run, which I think is what we saw at the end of the last talk. You kind of just normal clustering in normal single cell RNA sequencing clustering. But then because each of those beads also has a spatial location, you then get an image of your tissue. And the cool thing about this is that from the perspective of the end user, there's no microscopy involved, right? This is also, I should say, this is kind of fun because this is the first time that I feel like I'm telling this story from a historical perspective, because we're seeing lots of other technologies in the same, lots of other newer technologies at the same time. And so, the cool thing that we found, which now, of course, lots of other people have replicated with their technologies, is that these kinds of methods actually retain single cell resolution. So you're able to go in, for example, and visualize, in yellow here in this coronal section of the mouse brain. In yellow is the habenula, and green is this. Is the ventricles in the mouse brain. And we were actually able to visualize in blue here the single cell layer of ependymal cells between the habenula and the ventricle, which was the first indication for us that this method would really get us down to single cell resolution. And later analysis also that we did showed that actually not only that, but in the case of Slide-seq, 70% of the beads look like they have RNA from a single cell type. 30% look like they have RNA from multiple cell types. And again, that's something that you can fix by making the beads small. So since the publication of the original paper, I think one of the most important things that happened was that we did publish a Slide-seq v2, or I should say actually that my co-authors published Slide-seq v2 because by that time I had already moved on and was getting ready. This is December 2020. I was getting ready to start my lab at the Crick. Basically, the most important thing probably in Slide-seq v2 is the fact that there were some optimizations to the capture protocol, which meant that rather than capturing around 400 reads per cell, 300 transcripts per cell, we were able in Slide-seq v2 to capture something like 3,000 transcripts per cell. Okay, which is getting close to the kind of efficiency that you see in single-cell RNA sequencing. And I think actually, if you look at the like the different protocols that are now being published, Pixel-seq, Stereo-seq, I can't remember, seq-scope, you know, they all are kind of converging on this same, with Slide-seq v2, like people always present their data in a particular way to make their particular method look better. But I think that if you actually drill down into it, they all look like they're getting similar capture efficiencies. And so I think that this is really important to point out, because there's like a question as to what is the limit on this capture efficiency, because it's still, you know, much smaller than the number of transcripts that you see with like single molecule FISH, which is thought to be the gold standard in this area. You know, it's maybe not that much smaller. It's like 20% or 30%, right? But it's smaller, and yet everyone with like different modifications of the protocol seems to be getting roughly similar capture efficiencies. And so I think that there's an interesting problem to be solved there. But the cool thing, I mean, in the Slide-seq v2 paper, which I'd like to point out, is that now with the kind of capture efficiencies that we get, that you know, we get and that other people are getting, you know, we're now able to actually do things like look, for example, at like the profile of dendritic, of like RNA localization in like dendrites, for example, and like or another very cool thing that you're able to do, which is on the left and on the right here, is you know, combining RNA velocity with Slide-seq in the developing cortex. At which point you're like, because of this like phenomenon of cell migration, your RNA velocity becomes like a spatial velocity, which is, you know, becomes very cool. And you can actually see the migration of the cell types around the like radially, the excitatory cells radially, which is very cool. So yeah, so those are some exciting things, but I wanted to reserve some of the time. You know, fundamentally, my lab is like we're a lab of inventors, right? And so we're always trying to anticipate what's next. And so I want to retain a little bit of the time to talk about timestamps, which is another method that we just published recently, which might be, you know, the next thing to think about as people are starting to commercialize the spatial transcriptomic methods. Which the question here that we wanted to ask was like, okay, spatial transcriptomics is now a thing, but how do we get to the point where we're able to do temporal transcriptomics? And this is why, more generally at the beginning, when I said, you know, when I laid out what the lab works on, it was like contextual transcriptomics, right? So it's not just spatial context that's going to be important, but also temporal context. And so I'm just going to walk you guys through this pretty quickly. The paper came out in Nature Biotech, and this is a collaboration with Fei again, and with Ed and Linlin Chen. Linlin was an RA in Fei's lab who's absolutely brilliant, and she's now at Caltech doing her PhD. And she's a co-first author on this paper. So I think it was just really, I mean, she was really out of this world to get this co-first author position, basically like as an RA out of undergrad. So Linlin's great. So basically the idea here, and like the fundamental concept that you should take away, if you don't remember anything else about the temporal RNA sequencing, the fundamental concept that you should take away is that asking when a given transcript was transcribed, asking when a given transcript was made, is the same as asking how old that transcript is. Okay, and this is actually something that took us a long time to understand, and a long time to realize. But so I'm going to say it again. So like, if you want to know when a transcript was made from an endpoint measurement, right, you just need to ask how old is that transcript. And that's like entirely what timestamps is. So timestamps is a way of measuring the age of RNAs, which then allows you to infer when the RNAs were made, which then allows you to infer what the cell was doing at the time when the RNAs were made. And the way that this works is basically that we created an editing array. We're using RNA editing, right? So there are these enzymes, ADARs, which convert adenosines into inosines. And what we figured is that basically we could take an RNA and design like a cassette that you could stick into your RNA, which would have a bunch of adenosines on it. And then you attach this editor to that cassette, and the editor would start editing the adenosines and turning them into inosines, and inosines are interpreted by, you know, reverse transcriptases as guanosine. And so you basically see like this A to G editing in your subsequent sequencing. And by counting the number of those A to G edits, you would then be able to infer how old your transcript is, right? Which again then tells you when it was made and what the cell was doing, right? And so this is actually the like the kind of experimental system. So you have this, you have an RNA and you stick this thing into your RNA, and basically what you can see is that it's a bunch of a very AT-rich sequence with like some mismatches, because ADAR really likes these mismatches, and then it has these MS2 loops on the end, which are where the ADARs will attach. And if you go through and over time, I mean, basically the kind of experiment that you conduct is that you transfect a plasmid that encodes for, you know, an RNA under a given promoter into some cells. This is all like proof of concept stuff. I'll talk a little bit, you know, I'll talk in a minute about how you would actually implement this. But you have a plasmid that is expressing this like timestamp RNA. You put it in, you express some RNA, for example, if it's like in order to generate an artificial transcriptional event, basically we'd use a doxycycline dependent responsive promoter. You dox the cells, and then you add actinomycin D, which stops all transcription, and you let the cell, like you just leave the cells there, and kind of as over time the RNAs age, so you get to see what happens to the RNAs as they age, right? And you can just see these Gs accumulate in your sequencing. And when you go and you measure like at a population scale, like how many edits per, you know, what's like the average number of edits per RNA for RNAs that were made like a given amount of time ago, it's extremely reproducible. To the point where like we could identify, you know, if you gave us some cells and told us that these cells, you know, had a 1 hour burst of transcriptional activity some amount of time ago, we could tell you when it happened with like 5-minute precision, actually. Which obviously it's a little bit contrived because you would never be in a situation where you needed to know like, hey, we know it was exactly 1 hour, but like when did it happen, right? But the point is just that like this, from like a proof of concept perspective, this really convinced us that we're looking at technology that has, you know, we're looking at a phenomenon that has legs as a potential technology, right? And so then you can actually then go and say, like, imagine that you give me an RNA with a given number of edits. Can I infer how long ago that RNA was made? And the answer is yes, with about, you know, 3-hour time resolution, which like for an individual transcript doesn't seem like that much, right? So basically you tell me like, hey, Sam, this like RNA has, you know, 31 edits, when was it made? And I would go and I would look at this plot and tell you, oh, it was, looks like it was like, you know, somewhere between 7 and 9 hours ago, which like, that doesn't seem like 7 and 9 hours, it seems like a not that great from a time resolution perspective. But when you imagine this is actually like the first way that we're able to do this at like a, you know, transcript, you know, for like a population of different RNAs in the cell, right? So this is pretty cool. We're actually able to, for each RNA, go in and look at like how old is that RNA. But of course, you're never looking at individual RNAs. You're usually looking at populations of RNAs that are created at the same, you know, created like in an event. And then when you have a population of RNAs, then your time resolution, you get like improved time resolution because you get to do some statistics, right? And so the kind of experiment that we think about doing, you know, that we were able to do using this system would be to go in and say, imagine that we have 2 bursts of transcriptional activity, you know, that are some number of hours apart. So in this case, we had 2 different promoters. One of them was doxycycline dependent. One of them was light dependent. And you go in and you like, you know, induce with activity with doxycycline, then you induce with light, like 1 hour ago versus 5 hours ago. And this is the histogram of the number of edits you get. So in orange here is like or the transcripts that were generated by the doxycycline dependent promoter. Yellow is the transcripts dependent generated by the light dependent promoter. And so you get this editing distribution that has these 2 peaks. And you know, we then say, well, can you tell that has 2 peaks? And the answer is at a bulk level, yes, you can obviously tell. But the more interesting thing is if you downsample it to the case of like 50 RNAs or something, you know, something that would be reasonable to find in an individual cell, can you tell that it has 2 peaks? And the answer is actually like surprisingly, you can with like very high, you know, with pretty high certainty. I don't have time right now to go into like the algorithm that we built in order to do this, but basically we built an algorithm to infer transcriptional programs on the basis of like the number of edits per, you know, hit one of these histograms is the number of edits per RNA. And you find that you can like, you know, for experiments like this, where you have 2 different peaks of transcriptional activity, 2 distinct events, you're actually like very, very good at distinguishing those, that there were 2 events, even from a small number of RNAs. So like, obviously this is just like the first inklings of a of a fully temporally resolved transcriptomic method. Also, this is a method that relies on basically reporter RNAs. So like, you would build a reporter for a given promoter that is expressing one of these reporter RNAs, and then you look at those RNAs to tell you about when that promoter was active. Ultimately, we would like to go to whole transcriptome temporal RNA sequencing. So we're looking at we have a bunch of ideas about how to do that. I mean, again, if you're like want to do a postdoc and are interested in that kind of thing, please get in touch with me, because we would love to work with you on that kind of thing. So yeah, I think that's all I have time for right now. Just kind of the first inklings of some of those experiments. I want to thank everyone, both on the ticker tape. It's still called ticker tape up here. The original name was ticker tape, but on the timestamp side, and on the Slide-seq side, and then all the many funders who I've been very lucky to be funded by. And yeah, happy to take any questions.